So for a number of years now, we've had a tradition led by Chris Johnson, director of the SCI Institute, Scientific Computing and Imaging Institute at the University of Utah, which is not only our favorite visualization place in the world, but U.S. News and World Report keeps voting it the best in the world as well, so we're good on that together. And that is to use, frankly, it's the beauty of what you're doing, but also from a purely scientific basis, the ability of the human mind to understand through visual things. And this team and all the members you've brought, Chuck Hansen today, and over time, to help us visualize everything from outer space to very small biological structures, I just find it wonderful. And I'm glad we're doing this together again. I hope we keep doing it together again. And so today we're going to talk about a number of things, including that thing, and the idea of and I'd like to go kind of deep on this as we do it, but the idea of how our brains understand data, and that's the business you guys are in, is helping our brains to understand very complex data in ways which otherwise we couldn't do. And I think specifically we're going to talk about scalability and how to use the tools that you have perfected from a very, very small scale to a very, very large scale.
That's right.
Maybe with that, I'll turn it over to Chris. Okay.
Thanks, Mark.
Sure.
And good morning, everybody. Today, I have brought our most famous visualization researcher, Chuck Hansen. Every year, I ask Chuck first if he would come to FiRe and talk with me on the panel. And every year previous, Chuck has been engaged elsewhere in the world giving talks. But this year, you all luck out. Because he won't tell you, Chuck is the only one in the world who has received both the IEEE Visualization Technical Achievement Award and the IEEE Career Award, Visualization Career Award. So he is internationally renowned, and you will see why in a minute. We are very visual creatures. More than half of our brains are devoted to visual processing, unlike whales, which I learned at a previous FiRe conference, where 80% of the whale's brain are devoted to audio. So if we can find those right abstractions and mappings from this data, this often complicated, large-scale, complex data, and make those mappings into something that we can understand visually, that's the highest throughput to our brains. And so that's what we spend a lot of time doing, is trying to figure out either new abstractions and mappings, or to take current abstractions and make them scale to larger scale, more complex data. And so when we saw one of your themes, which was this multi-scale from the very small to the very large, I thought I'd give you a little bit of an overview of what we have done from visualizing the very small to the very large. And since we saw that Dennis was going to be the keynote speaker, we had to tell you about one of our newest, most exciting projects in space visualization. So we're going to go. All right. So this is part of the Connectome Project. Brian Jones said something about this last year at FiRe. We're working on trying to visualize this very, very large scale set of data. So what this is a project where they're using mosaicked electron microscopy to try and infer the fundamental wiring diagram of our brain. So each of these neuroscientists throughout the world are taking different parts of the brain, using this technology where they take the smallest piece of tissue, image it at the highest resolution of an electron microscope, dump the data, slice, image, dump, 24 hours a day, 7 days a week, until they have a view of the brain, and then they have to go through and track these individual neurons. And the idea is that everybody's going to do that, and when they're done, they'll be able to put all these different pieces together, and we'll have a three-dimensional visualization of our brains.
A wiring diagram.
A wiring diagram. So this, what you're seeing up there, is one mosaic slice. And to give you a sense, that little blue square is the highest resolution of an electron microscope. So that's 4,096 pixels by 4,096 pixels. And most of you probably still have HD TVs at home, which are 2K by 1K. I have a 4K projector. You should all get them. They're very nice. So that's about the new 4K projector. And as you can see, it's scaled through that. So once they get done with that, it's about 150,000 pixels by 150,000 pixels at a resolution of 2 nanometers per pixel. 2 times 10 to the minus 9th meters per pixel. So that's how small this is. And then they do the next slice and the next slice. They cannot use the same technology going into the depth. So we get this exquisite visualization and resolution across the plane, but even though they're trying to slice as thinly as they possibly can, you can see that in a very short distance, it's changing quite a bit. The other challenge that we have is the size. So one of these very, very small cubes of brain tissue ends up being 16.5 terabytes in size. And then they want us to automatically track through this data to come up with that wiring diagram. So one aspect of our research is to be able to interactively visualize 16.5 terabytes of data, which we can do. This is a visualization of that 16.5 terabytes of data on our, we call it a Powerwall, which is 36 30-inch monitors all put together that gives us 41 megapixels of resolution. And the researchers, the neuroscientists, spent hours and days looking at this data because no one had ever seen this data at this resolution. It was like a new type of microscope for them. And they discovered new structures, and they published. Now they want to be able to take this as input data and start asking hypotheses and doing simulations where 16.5 terabytes is now your input data set. Just to give you a sense, this is just one data cell type that's in this data set. Once you put in all the cell types, there's basically no space within there. So if you think about when people say our brains are highly connected, this is 2 nanometer per pixel resolution. This will give you a real sense of the structure at which we're so highly connected. Moving on to this from other cells. So this is on the 3D structural understanding and visualization of our anatomy and the brain anatomy. At the same time, what's going to be very important are more abstract, high dimensional resolution, high dimensional characterizations of biological data where we can infer understanding of how things go that we are not necessarily looking for a 3-dimensional spatial structure. So this is some work by Alex Lex, who's been here before, and this is a visualization of some genomics data for cancer subtype characterization. And so here is really trying to understand patterns and correlations and being able to do hypothesis testing.
And what type of cancer was that, or is there several?
There are, I don't remember what type of cancer this one is but I do know this is a glioma type of brain cancer. This is another one of Alex's work that he's trying to understand biological pathways within cancer in order to both understand and infer the underlying genetics and physiology, but also to be able to help design new drugs. And this particular system is being used by a major drug company to design new cancer drugs. And I want to put in a plug that I'm very excited to be being able to co-lead with Mary Beckerly, who is the director of the Huntsman Cancer Institute at the University of Utah, who is here today in a new Huntsman Cancer Institute, Scientific Computing and Imaging Institute, Computational Oncology Research Initiative.
Nice.
And so we are getting together to take the best of what we do at SCI and the best of what she does at HCI and see if we can make some real progress on this disease. And Mary's she'll be in a panel tomorrow at 1:30 too. And so very excited about that.
So we should talk more about that later.
We should then. Okay. It's one of my most exciting things that we're doing.
All
right. Now we'll move up to tissues. This is some work that Chuck has done, and he's going to tell you more about this. But this came from a real need from a biologist who was using these tools that just were not being able to cut it in trying to understand developmental and evolutionary biology. And Chuck was able to create this system called FlowRender, and it is really starting to make waves in the biological community from all these biologists using confocal microscopy. And we'll give you a preview here of what this is about.
FlowRender is a tool that was developed for confocal microscopy data, and it's focused on giving biologists an easy-to-use method for doing visualization and analysis of 3D confocal microscopy data. So Gabrielle Cardin here at the University of Utah has found that FlowRender is a valuable tool for her research in the Cardin lab.
The Cardin lab works on congenital defects in muscle development, such as congenital diaphragmatic hernias. We're using it to image developing limbs and developing diaphragms, which are very complex three-dimensional structures. So I would say that we would be unable to do any of our work unless we had FlowRender.
That's what we always like to hear from our collaborators. All right, let's move from neurons to cells to tissues to space. So this is a recent project that we have that's funded by NSF with some folks at the National Radio Astronomy Observatory. And it's looking at some of the data that's being collected by the Atacama Large Millimeter Array in Chile, the so-called Amo Array. And what you're seeing here is the raw data that it collects. And these are massive data sets. So they're collecting terabytes of data now on a daily basis. They'll be up into petabytes in the near future. And what we have learned by working with these astronomers and astrophysicists is that astronomy has really become a data science. Yeah. It's just overwhelmed by data. And a lot of it is very noisy data like this. And so when you look at this, what do you see? Well, I see a lot of little blue and kind of orange and whatever dots in there. And not a lot jumps out at me in terms of structure. But we are working with a team, so Paul Rosen and Bei Wang, our computer scientists. Bei has been here before. Seth and Betsy are physicists and astronomers, and working together to bring new tools for analysis and visualization to be able to better understand and take this data and come up with some underlying structure. And so this is using a topological analysis technique that Bei Wang created in order to be able to both filter out the noise in the data, but also be able to find structure in the data. And what this is actually you're seeing some what we think is rotation of dust around a black hole. And so we now have these tool sets that astronomers and physicists can use, astrophysicists can use, to be able to go into this very large scale millimeter wavelength types of radio astronomy data to be able to look for their structure.
Are you using visual computing techniques to do that?
Yeah, absolutely. Yes. So we have a new paper on this that's just been submitted to be able to use visual computing techniques to be able to do this and find patterns in the data.
Now that's the cool thing.
It is cool.
Okay.
So I think one of our most exciting projects is this one that Chuck is the Utah lead. It's an international collaboration that's funded by NASA, and it's called OpenSpace. What you see here is Gene Payne, who's one of the software developers, working on a multi-touch table, which we brought with us and is out in the foyer that we're going to have people use to show how easy it is to work around and taking in NASA data and other data sets. This is the entire Sloan Digital Sky Survey that's been brought in recently to the OpenSpace framework that Gene is showing you how to move about here. And what we'll do is be able to pop this on the Powerwall as well, and as Chuck will tell you, we can put this up on planetariums and other devices as well. So I think this is a really, really exciting project for us. And I'm going to turn this over to Chuck now so he can tell you more about it.
Before we do the jump cut, just so everyone gets this. So people are probably used to seeing mock-ups, models, things that look like that, and they're told, here is space, But what you're saying is this is the Sloan survey. This is actually
the real data.
This is the real data.
Yeah.
And thank you both. You have a table for us to play with. There's a little toy outside, and Gene is there, I think, as well.
Yeah, that's right.
To guide us, and we can, any of you can take a trip throughout the entire known galaxy or universe over there by the coffee stand.
That's right.
And so
that is right.
That's an amazing thing, and I'm really pleased you were able to bring that to us.
We are too.
I hope you all take a moment and do that. Fly to the furthest star you want to pick, or go through a black hole or something. That's amazing.
We have to have NASA data for it before you can
we have to have NASA data for it. So this is a NASA project, And I'll mention one more thing, which is that Andy Connolly was here with us last year from the University of Washington, and his team at Dirac Institute is about to be getting data like you're describing, perhaps even more from the same country. Of course, it's all in Chile, right?
Yep.
And I think in addition to the petabytes per day, instead of the Sloan survey, which I believe took like 10 years to do, it's every 2 days.
Yeah, it's amazing.
So what we're doing here, complete sky survey every 2 days, and trying to handle all of the pipeline of data coming down.
It's enormous
in that time frame.
And it's great.
Yeah, so the project is working with the Gaia Star Survey, which is this next survey that's coming on. We have prototypes of bringing that data in and for doing analysis, not just for displaying it, but for understanding the astroscience that it goes on. So let me Chris talked for a long time, so let me quickly go through this. FlowRender is an interesting tool because we developed it with biologists. I have an expertise in volumetric rendering, and they came to me and said, you know, we're really interested in looking at cell development over a time. Well, that's easy. You just do confocal scans over time. But what they want to do is look where individual cells go over time. So as organs are formed, such as the eye in the zebrafish, where do those cells come from? How do they go through mitosis? How do they move to form organs? And so we built a tool for doing that. And part of doing that is to come up with ways of doing interactions without too much trouble. So this was a tool that we built to do morphological diffusion segmentation. The problem with confocal microscopy, unlike MRI or CT, is that the light's attenuated as it's going through the cell, and so you don't get the same response for the same structure, the same biological structure. So we had to build tools that would allow people to pull these structures out easily by a painting on them. And by doing that, we have allowed people to come up with ways. So this is work from collaborative artists at the University of Tokyo, Kei Ito, who was looking at neural structures of the Drosophila brain, the fruit fly brain, and he extracted 96 different neural structures to build an atlas with it. No other tool that looks at confocal data could handle so many channels. So we came up with a way of doing it. Chris mentioned the Cardon lab. This was something that Gab's lab came up with. They were looking at the ulnar memory syndrome, and it was known that the bones were deiform, but wasn't known was the 40 different connective tissues that connect the bones to muscle structures, tendons, nerves, muscles themselves. And what she found through the process of knocking genes out of mice, which Utah is well known for, is that the TBX transcription factor, if it wasn't there, you would wind up with muscles that would not be formed. So we can look at a control mouse that has the full muscle structure and a transgenic mice in development that has structures missing. So this was exciting. This was something known that wasn't known before, that she hypothesized, built an experiment, used our tool to discover these differences, which is fun. So this is down from the tissue level, and we're looking at from that to the much bigger project. So OpenSpace is a NASA-funded project headed by the Hayden Planetarium at the American Museum of Natural History that includes partners of the University of Utah and NYU and Nora Scherping University in Sweden. And it's to go from what NASA is doing, what space is, to how NASA runs missions and how NASA processes that. So it's an open source project. It's used for public dissemination. So in the middle you see a planetarium presentation.
instrumentation. It can be used as a research platform for visualization. So on the right, you see the Illuminati portable dome for looking at this. And it can be used as science. So on the left, you see the magnetosphere coronal mass ejection from the sun that's done with NASA Goddard, the community coordinated modeling system. So OpenSpace is built around this geometry system that NASA built called SPICE, which all of us that read Dune love this acronym. And it allows us to look at things that we haven't seen before. So here's an example of Pluto, where we started with low resolution telescopes, moved through higher resolution telescopes to the most current imagery that we have that was taken from the New Horizons mission, which passed by Pluto in 2015. So on the left, you see the spacecraft itself modeled in OpenSpace with the different instruments that it has. And what SPICE allows us to do is, in a space-time domain, understand where that spacecraft is at some given point in time, what the attitude of that spacecraft is at any given point in time, and which instruments are active. And by doing that, we can project the frustum of the instruments into space to see what they are sensing. So at the upper right, you can see the Pluto sensing as it's going on, and down on the lower right is as New Horizons was a ways away from it, we got a low-res image, and as it flew closer, we could snap high-res images. Now, when the spacecraft was flying, we didn't have the data, of course, because that data takes quite a while to come across the solar system back to Earth and be processed. So it was just these green squares that were going on. And the morning that this took place, there was a breakfast with Pluto that was hosted by Neil deGrasse Tyson, linked up planetariums in North America, South America, Europe, Asia, and mission control. And they got very excited seeing this because they could see what the spacecraft was doing as it was doing it, not see what the actual data was.
Wow, I'm tongue-tied.
I don't know what that is, but would you turn it off?
It's a video.
Oh, I'm sorry.
Yeah.
It's a video. So sorry.
Yes.
This was mission control laughing and getting excited because they were seeing the spacecraft image Pluto as it was taking place, and they said, "We can't believe this is really taking place. This is really going on," which is fun. But it also has planetary mapping capabilities. Let me start this video playing if I can, where using data provided by NASA, both for color layers, height field layers, overlay layers, we can look at data on planets. So this is looking at the planet Earth, the daily cloud image that's provided by the NASA Gibbs survey. And we can also look at things such as the surface of Mars, which first NASA flew through with a Viking many decades ago and took color images from it. And then they sent up the Mars Global Survey, which took height field data, that is terrain data, elevation data at a low resolution. And in 2007, they launched the Mars Reconnaissance Observer, which has several different instruments on them, including CTX, which is a contextual grayscale imaging, but also a high-rise camera, which the USGS can process stereo pairs to get terrain data. So what we're seeing here is a fly through Mars using the high-rise data, the CTX contextual data with the Viking colors. The path is the Curiosity rover, which also has stereo cameras, and we can process those stereo cameras to look at what the rover's seeing and get higher definition overlaid onto the high-rise image
to get a better sense of what the surface of Mars is doing. And as we zoom in, you can see that you get very detailed geometry of what the terrain is. And this is all processed with data provided by NASA from services, web services that we brought into OpenSpace, which is good. The Carrington event was this geomagnetic storm that happened back in the 19th centuries. The northern lights were seen as far south as the Caribbean. The telegraphs in Europe and America were shut down. They completely failed. If the same type event happens in 2019, it's estimated that the economic cost would be in the trillions of dollars. The satellites would stop working. Your car would stop working. Your cell phone would not work. The telephone system, the grids would all go down. Your coffee pot wouldn't make coffee in the morning. It would be a disaster, right? So it isn't good, but fortunately for us, whoops, let me try to get this going. There we go. The Community Coordinated Modeling System at NASA Goddard looks at coronal mass ejections from the sun with satellites, feeds that into computational models that they have to predict where solar storms are going using these different satellites to feed into a model to make daily predictions, and they're using OpenSpace to help them understand the 3D spatial relationships with these magnetic storms. So as Chris pointed out, we brought our touch table up here, and we're lucky today that Alex Bach, the chief software architect for OpenSpace, who's a Sloan Data Science fellow, and Sloan Data Science is having their meeting in Park City tomorrow and Friday. So he happens to be here today. So he'll be working with Gene to explain the touch table and help guide you through things. And OpenSpace is an open source software project. There's a website that explains more about it. And of course, if you have questions, you can feel free to send them to me.
Maybe one question from the audience if somebody wants to step up, and I have one question, which is that really kind of scary thing you landed on at the end, the return of the Carrington event. How much time warning would we have with all of this predictive monitoring?
So as the solar coronal mass ejections take place, we have on the order of days before the storm's going to hit. The problem is they can predict it and warn us about it, but taking some action into effect, we don't know.
Days wouldn't be.
There's nothing. You can shut things down, power things off,
turn off the grid,
and hope that that's going to resolve it. But that's the current solution. Thank goodness this hasn't happened in the past century, but it's going to at some point in the future.
Do we have any way of knowing how many other Carrington events there were before and what kind of frequency they happened with?
Unfortunately, the history before the 19th century didn't really record those, and so there's not a data point as to when this is going to happen.
Could be.
The sun constantly is going through coronal mass ejections, sending these solar storms through space. It's just that they have to come through space as the Earth's rotating through. So it's a time-space.
In other words, we don't know.
We don't know.
Okay.
And I hope it doesn't happen in my lifetime.
I do too.
So the table will be here all day today. We're going to have to take it back later today around 5 or so. So please take an opportunity to talk with Gene. And besides being able to look at some of these missions, like you can walk around on Mars, et cetera, you can also look at events like the big eclipse we had last year. And so he can go back in time and look at previous events. So take advantage of that, and we hope you enjoy that.
Very cool. Well, we've just crossed over to red, so thank you.
Okay.